Alrighty, so we're learning more about this stuff called molecular biology. And now we know that DNA does two different things. DNA is involved in replication. DNA is involved here in replication. And DNA is involved here also at the first step right here, transcription. Okay? Transcription will make RNA, and then it's going to be RNA that is involved in translation. And of course, proteins are here all the darn time. So we talked about DNA replication quite a bit, and we know the essentials, the Gattaca stuff. Let's talk a little bit more about transcription and translation. That did not go well. Translation. Whenever you think of translation, you think, someone translates from one language to another, or someone can translate from code into what is the message, right? What's actually message. Hello, the message. And so I write the difference between translation and transcription, because translation is translating the code. You're going to hand me, as a ribosome, you're going to hand me messenger RNA. Messenger RNA is still in the form of code. It's in RNA nucleotides, but it's still in code. Instead of G's and A's and U's and C's, it is going to be G's and C's and U's and A's, right? So it's just going to be, but still in code. It still doesn't tell me what amino acid it is. In order to know what amino acid, we need to translate it. This was going to be the DNA. We turned it into the messenger RNA. And now that ribosome needs to read that and build the protein. And he does that in the process called translation. So like I said, you can think of transcription as the process by which messenger RNA gets made, and then and that messenger RNA leaves the nucleus, and then translation will be the act by which the protein gets made using the instructions that are found still in code in the messenger RNA. Now, for translation, we need three different types of RNA. The first type of RNA we've already been talking about a ton, messenger RNA. Messenger RNA carries this coded message, right? Still no one knows what it means, but it's still in code, but it's messenger RNA. So when the nucleus is in charge, I want more of this protein built, then the way it does that is it sends out messenger RNA, and it's up to all the machines out there in the cytoplasm to obey that instruction by building whatever they have, whatever the nucleus has told them to build. Transfer RNA. We will talk more about transfer RNA. However, it is going to deliver the proper amino acid to the growing protein chain. So its job is transferring. Transferring what? Transferring an amino acid. Remember, this is going to be in code. Let's just spell cat a bunch of times. Okay. This is going to be the DNA code, and then we made messenger RNA. And then each one of these is going to mean bring a certain amino acid to the growing protein chain. So whatever that amino acid is going to be, say it's going to be the letter P, okay? Now, the letter P represents an amino acid. Nucleotides, CA, etc., those are the monomers that make up the polymers that are DNA and RNA. Amino acids, that's these guys in purple, they're the monomers that make up a protein. So the job of the ribosome is one amino acid at a time. It's going to put them together to build the protein. You probably learned that amino acids put together in the right sequence, that is the primary structure of a protein, which dictates secondary, which dictates tertiary, etc. How does the ribosome know what order to put them in? The ribosome will simply pause at each codon, and then the transfer RNA that matches that codon will bring the right amino acid to the chain. We'll talk a little bit more about that. Then we've got ribosomal RNA, and ribosomal RNA is the easiest one because ribosomes are made out of ribosomal RNA and protein because protein does everything. This is what a transfer RNA looks like. Every transfer RNA carries an amino acid. Which one? Each transfer RNA carries only one kind of amino acid. If this is the transfer RNA that carries alanine, it will never carry another amino acid, only the amino acid alanine. Now, this is important, so pay attention. This area right down here is called the anticodon. It is called the anticodon, right? Let's go back one. If this is the codon on the messenger RNA, GUA, 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 and if this anticodon, if this codon, GUA, if it is meant, put a proline here, then the proline, or whatever amino acid, we'll say amino acid X, would be attached to, that's supposed to be a transfer RNA, whose anticodon is CAU, right? Notice that the anticodon matches, is complementary to the codon, right? A couple of things. Transfer RNAs always have an anticodon. Transfer RNAs do not have codons. Codons are little code words that are a part of a coded message, okay? Transfer RNAs are built out of a lot of nucleotides. You could put them together 3 in a row, but those are not codons. It's not a codon because it's not a part of a message. Anticodons are found only in transfer RNAs, and they are going to be the complement to a codon in the messenger RNA. And if the anticodon matches the codon that says put a methionine here, then the amino acid that it carries will be methionine, right? Okay, anticodons. So DNA transcription takes the DNA blueprint and makes a little RNA version called messenger RNA. DNA stays in the nucleus. This is going to leave the nucleus. It leaves the nucleus through a nuclear pore. And then in translation, this mRNA that left the nucleus, it is going to be used as the instruction manual. And the ribosome is going to read it one codon at a time. And then transfer RNAs are going to bring one amino acid at a time. And the job of the ribosome will be to take the first amino acid and attach it to the second amino acid, and slowly, one amino acid at a time, grow the protein. This whole thing that's in baby blue, that is the ribosome. It is made out of ribosomal RNA. Okay? The thing that is in yellow, that is transfer RNA. And you can see right here, that is going to be 3 nucleotides in a codon on the messenger RNA. And right there, that is the anticodon that matches the codon in the messenger RNA. This particular transfer RNA just brought that guy to the ribosome. Why are there so many amino acids? Because it was up to the ribosome to take all of them that had already been built and attach them here. And now when the next transfer RNA comes in, this whole thing will be added to that one, et cetera, right? DNA is meant to be permanent. RNA is meant to be temporary. DNA is stabilized by the DNA double helix, and it's also protected and organized by proteins. RNA is meant to be temporary. Our ribosomal RNA, that's what makes up the ribosome, the tan stuff here. Transfer RNA, that's this blue guy here. Transfer RNAs have an anticodon. See that one? UAC. See how it is the complement for the codon on the messenger RNA? That's how it works. And pink is going to be the messenger RNA. Now, one of the things you're going to have to do on my quiz is to be able to take either DNA that I give you or messenger RNA that I give you and tell me the sequence of amino acids that is in that coded message. All right? So pay attention. If I give you DNA, then you need to transcribe it into messenger RNA. Once you've got the messenger RNA, then you will use this table because this is a messenger RNA table. Now, if, on the other hand, I give you messenger RNA, go straight to the table, right? Happens all the time that students are like, they get messenger RNA. If I give you messenger RNA, don't transcribe it. It's already been transcribed. If you transcribe it, what you're doing is you're figuring out what all the anticodons are. This is not a codon for this is not a table for anticodons, right? All right, so if I give you DNA, and you'd know it was DNA because it's got a T in it, but I'll tell you anyway. If I give you DNA, the first thing you have to do is transcribe it. So instead of TCG, it'd be AGC. Great, okay? So the first letter of the codon is A. All of the, oh, you know what I didn't tell you? Probably your lecture instructor did, but if you take 4 letters, G-A-T and C or G-A-U and C, if you take 4 letters and put them into 3-letter combinations, you can make 64 unique combinations, like UUU, UUC, UUA, UUG. Please stop me, right? And this is all 64 of them put together in a table. And here's how you read it. You have got this codon AGC. You start with the first letter A. Okay? Everything that's in that box that is now yellow, all of those codons start with the letter A. All of these guys start with the letters A, U. All of these guys start with the letters A, C. All of these guys start with the letters A, right? You need AG. All of these guys that are here, they start with AG, the box in green. Great. We're almost there. We need to find AGC. So we go to the third letter, AGC. Boom. The answer is serine. Got to be able to do that. Now, if I gave you AGC and asked you this question, you would go straight here. So please don't do this. Don't get messenger RNA and then turn it into UCG, because then you would get UCG. Oh, you'd also get serine. You'd still get the right answer. Well, that's strange. Actually, it's not strange. It's funny that I figured that out just now. But I told you that life needed to solve the problem of how to not make mistakes, how to catch mistakes and fix them before they become permanent mutations. And the third problem was how to minimize the effect of mutations. I'm going to start with that at the beginning of our next video, and it'll all come down to this.